I'm Kate Speak. I'm a research faculty member at the Benaroya Research Institute at Virginia Mason in Seattle, Washington. The work I'll describe today was a highly collaborative effort involving investigators at 7 different institutions who all worked together on shared sample sets from the Immune Tolerance Network, or ITN, and with funding from JDRF. Type 1 diabetes results from the immune destruction of beta cells in the pancreatic islets, and it leads to an inability to produce sufficient insulin. So here I'm showing the data for the subjects included in the study you'll see today. These data all come from samples from people who were randomized to the control arm of new onset type 1 diabetes clinical trials. And what that lets us do is to look at the natural history of the disease right after diagnosis in the setting of these very well-characterized subjects and samples. The subjects were all enrolled within about 3 months of diagnosis, and that's the point that you can see there on the left, with the insulin secretion levels at diagnosis shown on the y-axis. So most people with type 1 diabetes are actually still making some amount of insulin at diagnosis, but you can see when we connect the dots that insulin secretion declines in general over the course of the first couple of years after diagnosis. However, this also highlights that people have very different clinical courses in the early years after they're diagnosed. So for example, the subject in blue here has no change in their insulin secretion over 2 years, but the one shown in olive stops making any insulin even before the end of the study. Now, in part, this is driven by baseline insulin secretion, and it's been shown previously many times that the amount of insulin that you make at diagnosis will actually correlate with how quickly you lose your insulin secretion over time. But this also brings up a really important key point, which is that age also plays an important role. So the subject in blue was diagnosed as an adult, but the one in olive was diagnosed as a young child. And this age difference is also known to play a role in decline in insulin secretion. However, age is a very imperfect predictor, as the subjects that are in purple and salmon highlight. So these guys were both diagnosed in their late teens, but they show very different rates of decline. And all of this is really important for a couple of reasons. So first, we know that maintaining some insulin secretion can be protective against future complications of the disease. And second, because it would be really helpful to understand in advance what someone's profile is going to look like in order to identify those that are most likely to show a decline in insulin secretion, and thus who are most likely to benefit from therapy in a clinical trial. So the main question that we had was a practical one. Could we identify immune markers at the time of diagnosis that were associated with this sort of future change in insulin secretion? To do this, we employed 2 key methods. First, the selection and validation of a dozen different assays. We picked assays that were thought to provide independent information, and we included both those that were mechanistically related to type 1 diabetes pathogenesis, like proinsulin C-peptide ratio, as well as assays that could generate sort of more broad-based hypothesis-generating results, like RNA-seq of multiple immune cell subsets. We then validated those assays to ensure that they were providing robust and reproducible data. The next key method was the development of a novel analytical tool that could combine data across both single analyte assays and omics scale assays, and then could let us identify a composite model rather than a single analyte that was associated with C-peptide decline in our subjects. So the first step in our process was to test the reproducibility of each of the 12 assays that were included in this project. The overall goal with this was to reduce the likelihood of type 1 error by assessing how robust every single analyte and every assay was, and then removing those that didn't perform to standard, which sounds a little bit boring, but it's totally critical to this work. And in fact, the study was conducted through the JDRF Core for Assay Validation, which I co-lead with Alice Long at BRI, and which was established to let investigators who study type 1 diabetes get samples and resources that they might need to assure assay reproducibility throughout the field. So here what you can see is all the assays that we selected for the study. Through this process, we removed 2 assays and thousands of analytes from the subsequent phase of the study. However, over 75,000 different analytes still remained, which we then measured in the cohort of new onset type 1 diabetes subjects for whom I showed the insulin secretion data earlier. So to combine and filter the data from those subjects, we developed a new analytical tool called DeFacto, in collaboration between BRI's bioinformatic team and Rafael Gottardo's laboratory at the Hutch. Now this tool, as I mentioned, needed to be able to bring many different data types together. So the first step in the analytical process is the merging and scaling of the individual assay data sets. Next, the tool conducts an initial filtering step. This filtering is customizable, We opted to choose only to keep 30 analytes per assay, which put all the assays on a more even playing field for later. Importantly, though, we also incorporate the clinical and demographic data at this stage. Next, the analytes from all assays are clustered, and a single representative analyte from each cluster is selected for further analysis. At this point, we've filtered the data down to dozens of analytes from the original genome-scaled datasets. Finally, the tool uses Lasso to actually select a model associated with decline in insulin secretion from among those dozens of analytes. So after all of that, what did we find? What we found was a panel of 12 analytes plus baseline insulin secretion that in combination were highly associated with decline in insulin secretion over 2 years. So here you can see each individual analyte that's a component of the model and its independent correlation to C-peptide. I also want to point out that we tested the tool in a separate published dataset to see whether we could find previously identified predictors of vaccine efficacy in an HIV vaccine trial. And we actually found the tool performed very well in that independent dataset too. So to summarize, our interest in the future is in applying these tools to studies in type 1 diabetes and to moving this panel of 12 analytes forward for further testing. And our hope is that the tool and this method of assessing reproducibility can both be useful to analyzing other datasets and looking at other disease outcomes. As I mentioned, many people have been involved in this work, and I truly appreciate all of their efforts, and I thank you for watching.